Okay, good evening everyone. Good evening. Good evening all. Okay. Okay, anyone else is going to join? Okay. So friends, today we are going to continue this particular plant physiology. Good evening, very good evening to all of you friends. Let's continue. As a part of the plant physiology friends, we have completely done the process of photosynthesis. Now what we are going to read friends, now we are going to read the next topic, that is the nitrogen metabolism. So friends, nitrogen metabolism is also one of the most important topic I can say for the CSIR NET exam. The advantage and disadvantage with this particular chapter is friends, I want all of you to remember the advantage or disadvantage with this particular chapter is the chapter is a very easy, the chapter is the subunit is very easy, but the disadvantage is that friends, information is more. You have to remember more amount of the information. Actually concept is small, information is more, and especially this kind of concepts, we often forget. So that's why we have to do multiple revisions while solving while learning this particular concepts. Okay. Friends, from this particular nitrogen metabolism, what kind of questions that we can expect? So friends, we can expect the questions from the processes. I will explain everything very clearly. Okay, good evening all. We will get the questions from the processes. What are friends, we will get the questions from the genes involved over there. And friends, we will get the questions from the enzymes involved in this particular nitrogen metabolism. Okay, then my friends, another important point, we can also get the questions from the bacteria also. So friends, there are many ways are there where we can expect this particular question, right? We can expect the question. And listen this PPT for the nitrogen metabolism. So friends, all of you listen to me. When I taught the photosynthesis, I have told you that the PPT will be 100% sufficient. So questions won't go beyond the PPT whatever we discussed. Now we are discussing about this particular nitrogen metabolism. Okay, so for this PPT I am talking, this will help you up to 95%. Okay, and I'm requesting all of you, once the lesson is over, I'm requesting all of you just revise the Pathfinder once. By that you can easily understand it. Okay, so it's a very easy topic, and our luck is we can expect a part B question also, my friends, and if our luck is really good, we can expect the part C question both. But 99% friends, this particular nitrogen metabolism chapter is the this sub concept is majorly important for the part C questions only. Okay, now let's start friends, and it's easy. It's the concept is not at all difficult, but what is only difficult is you have to remember maximum amount of the information. Little bigger concept it is, we will finish it in the 2 classes. So I'm requesting all of you friends, wait for 15 minutes, listen to me very carefully. I will cover every single point, pay attention, and after 15 minutes I'll solve all your doubts. So nitrogen cycle. So friends, everyone listen to my words. Can't we live without nitrogen? Of course I say we can't live without nitrogen. Friends, why can't we live without nitrogen? Because nitrogen is extremely important for the production of proteins, for the production of amino acid. You take an example of DNA also. What the DNA and RNA contains, they contain the nitrogenous base. Is it? Now you have to understand how important the particular nitrogen is. Everyone listen every single point whatever I am saying. But the point is here, where the nitrogen is present. Nitrogen is present in the atmosphere 78%. But friends, all of you listen my words very carefully. Whatever the nitrogen that is present in the atmosphere, friends, that particular nitrogen is the chemically inert. What do you call it this friends? Chemically inert. What do you mean by the word chemically inert? We can't use it. In other words, I can tell my friends, the nitrogen which is present in the atmosphere, friends, it is N triple bond N. So we eukaryotic organisms like plants, we eukaryotic organisms like animals, we can't break this particular N triple bond N. But listen to me, just now I said it to all of you very clearly, we can't live without nitrogen. We have to use the nitrogen compulsory. But what is the point? We can't use the nitrogen which is present in the atmosphere directly. So when can we use the nitrogen? We can use the nitrogen whenever the nitrogen is available in a simple form, whenever the nitrogen is available in a free form, then only we can use this particular nitrogen. What are the free forms of nitrogen? I can say NO2, I can say NO3, I can say NH3, I can say NH4 plus. These are all what? Simple forms of nitrogen. Listen to me, I will cover every single pinpoint. Focus. So this is what? Simple forms of nitrogen. We eukaryotic organisms like plants and animals, we can use the nitrogen when it is in this forms only. But do you think we will convert this atmospheric nitrogen into this simple forms? We can't do that because we don't have an enzyme. Now who is going to do it? So friends, the atmospheric nitrogen can be converted into most simple forms by what friends? Bacteria only. Now I want every one of you look into this particular diagram, friends. Every one of you pay attention towards this diagram. Pay attention. So friends, see here, this is the overview of the lesson what we have to read. See here, atmospheric nitrogen, first we are going to be converted into NH3 or NH4. What is this process? We call it as a nitrogen fixation. I told you, right, we can't use the nitrogen directly, we have to convert into simple forms. I'll explain, just listen. So that's why we converted into NH3 or NH4. This is what do we call nitrogen fixation. But friends, once again we are converting NH3 and NH4 back next to the NO2 and NO3 because they are more simple forms. This is what do you called? We called nitrification. Friends, don't get confused. Conversion of NH3, NH4 to NO2 and NO2 to NO3. The entire process we will be calling it as the nitrification only. Do not forget that. Okay, so friends, once again I am reminding to you why I have converted N2 to NH3, NH4. Why I have converted NH3, NH4 to NO2 and NO3. Why I have done all this? To make use of it, to utilize it, to incorporate in the body, or to assimilate in the body for the production of proteins and amino acids. Now my friends, this particular nitrates get assimilated into the plant. Now the plant produce their proteins, their amino acid. Upon eating the plant, who will get? Animals will get. Now animals also getting benefited, nothing but we. Now friends, after the death of the plant, after the death of the animal, what is happening? The dead organic matter of plant and animal can be decomposed by the decomposers. Then decomposers also will take the nitrogen because they also require nitrogen. Then excess amount of the nitrogen they fixed in the soil in the form of ammonia, which we called as ammonification. Now my friends, everyone pay attention. Plant got benefited, animal also got benefited, decomposers also get benefited. But from where we have taken the nitrogen? We have taken the nitrogen from the atmosphere, is it? That's why my friends, we have to send the nitrogen back to the atmosphere. So NO3 can be converted back to the N2, which complete the nitrogen cycle. This process only we called denitrification. So all of you listen, how can I define the word nitrogen cycle in a one line? The cyclic movement, listen my friends, the cyclic movement of the nitrogen from one tropical level to the another tropical level throughout different levels or throughout the cycle, which we called nitrogen cycle. Simple. The cyclic movement of nitrogen from one tropic level to the another tropic level through the group of organisms. we call it as the nitrogen cycle. So every single process we have to read very briefly and very elaboratively. As I said to all of you, PPT will help you 95%. So I want every student has to understand every single pinpoint. Now, what are the processes that we have to study? These are all the processes: nitrogen fixation, nitrification, assimilation, denitrification. Then friends, followed by the NMOX process. Sometimes even we got the question also, not in this particular plant physiology chapter, the same thing they have asked in the ecology actually. In same processes they can give in one column, and friends, in another column they just give the equation. So friends, how can we write the nitrogen fixation? I told you very clearly, right? Where N2 can be converted into what, friends? NH3 or NH4 plus. This is what exactly I will be calling it as the nitrogen fixation. And my friends, how can I define the word nitrification? I have clearly told you this particular NH3 or NH4 plus, I am converting into NO2 first, then NO2 I am converting into NO3. This entire process we called nitrification only. Then what is the next step? Assimilation. You can ask me, sir, why we have done all this? Why I have converted the nitrogen into most simple processes, most simple compounds? Why I did that? In order to incorporate in my body. So conversion of NO2, NO3 into amino acids and proteins. This is what do you called assimilation. Then friends, what do you mean by the word denitrification? Conversion of nitrate back to the atmospheric nitrogen, because we have taken the nitrogen from atmosphere, so we have to send it back compulsory. That's what we call denitrification. Then the last step, NMOX. My friends, I want every student has to remember, so far we did not identify many bacteria that are involved in this process of NMOX. So far friends, we have studied only 5 bacteria, just 5, that are involved in the NMOX process. What is NMOX? You just now read, right? We are converting NO3 back to the N2. But by using this particular NMOX bacteria, friends, we can convert NH3 directly to the N2. That is what do you call the NMOX process or the NMOX bacteria. I'm going to explain you step by step. Okay. Once again, before continuing, I am just saying to all of you, the concept of nitrogen metabolism is very small, but my friends, information is vast. Hence, I am requesting all of you, try to focus on every single slide, try to remember maximum amount of the information. Let's talk one by one step elaboratively. Friends, among all these 5 steps, which are very big steps, the nitrogen fixation is the very big step. At the same time, friends, assimilation is also very big steps. So let's focus one by one in a elaborative way. Nitrogen fixation. Listen to me for 15 minutes. Conversion of the atmospheric nitrogen to NH3 or NH4. This is what exactly I can call it as the nitrogen fixation. Friends, you all people know one point very commonly. We can fix the nitrogen in a different ways. You know, right, when the natural lightning comes, natural lightning also, friends, fix the nitrogen, but not to the greater percentage. Natural lightning fix the nitrogen up to 5 to 8%. Do not forget that. At the same time, friends, we can fix the nitrogen industrially also. We read in our +2 level by a process called as Haber's process. I hope every student remember. But my friends, this is not what we have to read, right? We have to read exclusively bacterial nitrogen fixation. Why we should read the industrial nitrogen fixation? We have to read how the bacteria is going to convert the atmospheric nitrogen into most simple forms. That is what we have to study, my friends. So who will do this particular bacterial nitrogen fixation? Bacteria. Every student has to remember the versatile enzyme. What is this versatile enzyme that is involved in the nitrogen fixation? Nitrogenase enzyme. You should not think that the nitrogenase enzyme present in my body. No, I don't have. Nitrogenase enzyme is exclusively present in the bacteria only. Nitrogenase enzyme not even present in the plants also. That's why plant can't fix the nitrogen. Animals, we humans can't fix the nitrogen. Only bacteria can fix the nitrogen because they are having a versatile enzyme, nitrogenase enzyme. Now let's try to understand elaboratively what are the different type of bacteria that are involved in this particular nitrogen fixation. Friends, all the bacteria which are involved in the nitrogen fixation, listen to me friends, broadly categorized into two types. One we called free living nitrogen fixation bacteria. Friends, next one we called symbiotic nitrogen fixing bacteria. What are the major differences between the free living and symbiotic nitrogen fixation bacteria? We read many times. Symbiotic bacteria are those type of bacteria, they always maintain a mutualistic relationship with the leguminaceae plants. So what is the point? Bacteria is involved in the nitrogen fixation also. But friends, when you look into the free living nitrogen fixing bacteria, what is the speciality you have to remember in the free living nitrogen fixing bacteria? They do not require anyone's support. They do not require anyone's support for their growth and develop. They grow on their own, they develop on their own. At the same time, they can fix the nitrogen on their own. That's why, my friends, we will be calling this as the free living nitrogen fixing bacteria. I don't know what you do, my friends. It is extremely important. I want every student has to by heart this particular chart. Here nothing to explain. There is a great chance we can expect our part C questions in the matching. Friends, if you really look into our 2022 February CSIR NET Life Sciences question paper, if you see very clearly, there are 13 questions were appeared in the matching. And all 13 questions are very, very easy questions. Hence, wherever there is a chance of getting matching questions, we should not leave. Now focus. Symbiotic nitrogen bacteria and free living nitrogen bacteria. Friends, it's easy, but the only point is you have to remember for the long time. See, please don't read. If you read, you will forget by tomorrow itself. Never read. Try to visualize it. Take the printout, paste it on the wall. Try to visualize it every time. Try to remember in the form of a image. Nitrogen fixing symbionts, all the rhizobiums like azorhizobium, brodyrhizobium, photorhizobium, cynorhizobium, these are all the rhizobium species, friends. So they are all living on what plant? Leguminaceae. Best example you can remember, groundnut. You can remember groundnut and you remember Parasaphonia. I want you to remember these two plants. Then there is a bacteria called Frankia. Please remember only this thing, Casuarina tree and alder tree. Just remember these two enough. Then Nostac living on the Gunnera plant, Anabina living on the Azolla water fern, Acetobacter living on the sugarcane. Nothing to explain over there, my friends. You have to remember that. Now, free living nitrogen fixing bacteria, they can live on their own, they can grow on their own, but they are having the ability or capability to fix the nitrogen. Let's take an example of cyanobacteria. What are cyanobacteria, friends? They are the blue-green algae. What are the best example? Anabina, Calothrix, Nostac. You don't leave a single example. Whichever I marked, never leave. Aerobic. What are aerobic bacteria? We all commonly know that aerobic bacteria are those type of bacteria compulsory require the presence of oxygen for their growth and development. Without oxygen, bacteria cannot survive. If the bacteria is unable to survive, how can they fix the nitrogen? If they want to survive first, they require the oxygen. So best examples are Azospirillum, Azotobacter, friends, Bezeranschia. Then facultative, we read it very clearly. What do you mean by the facultative? I told you many times. They can grow in the presence of oxygen. They can grow in the absence of oxygen. But my friends, when there is a presence of oxygen, their growth will be double. There will be a more growth in the presence of oxygen. But also there is a minimal growth in the absence of oxygen. Please remember Bacillus and Klebsiella. These are all the anaerobic bacteria. Some are non-photosynthetic, some are photosynthetic. Friends, let me tell you one interesting point here. What is that interesting point? If you look into this very clearly, friends, Nostac, Yanabina, this Acetobacter, these three are the bacteria which can fix the nitrogen symbiotically also, and which can fix the nitrogen by free living also. Please kindly do not forget that particular statement. They can ask you, they can ask us in a part B question stating that which of the following bacteria can act as a free living bacteria and symbiotic bacteria. Every student has to remember this, friends. Nostac, Anabina and Acetobacter. Do not forget. Friends, still we are reading the first step only. Nitrogen fixation. It's a very big step. Focus. So we understand what is the definition of nitrogen fixation. We understand that we can't do the nitrogen fixation. Plant can't do the nitrogen fixation. That's why bacteria are involved in the nitrogen fixation. Now all of you just listen to me very carefully, friends. From this particular slide, I am requesting every one of you understand. You have to understand more correctly. I have clearly said, listen to me, whether it is the symbiotic nitrogen bacteria, whether it is the free living nitrogen bacteria, doesn't matter. Both can fix the nitrogen. There is no doubt about it. Why I am saying they can fix the nitrogen? I am saying they can fix the nitrogen. The reason behind that, they are having an important enzyme. What do you call it, that particular enzyme? We will be calling it as the nitrogenase enzyme. See, all of you listen. I am a bacteria. In my body, what is there? Nitrogenase enzyme is there. That's why I can fix the nitrogen. Please continue, everyone. Now here comes an interesting point. I am a bacteria. In my body, nitrogenase enzyme is there. But friends, there is one complication, one disadvantage with the nitrogenase enzyme present in my body. What is the disadvantage with the nitrogenase enzyme present in my body? Nitrogenase enzyme will become immediately inactive if there is a presence of oxygen. Once again, listen to me carefully. I am a bacteria. Inside my body, nitrogenase enzyme is present. If I want to fix the nitrogen, I should always maintain what kind of condition I should maintain? I should always maintain the anaerobic condition. Why should I maintain all the time anaerobic condition? All the time in the sense whenever there is a fixing of nitrogen, because nitrogenase enzyme get inactivated by the oxygen. Now listen. How bacteria are going to maintain the anaerobic condition while fixing the nitrogen? I am telling you one important point. Listen, you should not think that, sir, does the bacteria will maintain the anaerobic condition all the time for the nitrogenase enzyme? No, not at all. That is wrong statement. All the bacteria are going to maintain the anaerobic conditions whenever nitrogenase enzyme wants to fix the nitrogen, only that time. Otherwise, they won't maintain the conditions. What are the one of the best anaerobic condition? We can call the heterocyst. So now you can ask me once again, sir, does the heterocyst will be formed every time? I say that no, it never form every time. The heterocyst is basically formed in the cyanobacteria, friends, whenever it is fixing the nitrogen. If it is not fixing the nitrogen, the heterocyst cannot be formed. Now you can ask me, sir, what is the function of heterocyst? First of all, what is our goal? Our goal is to maintain anaerobic condition. So to maintain the anaerobic condition, how the heterocyst will help us? All of you remember, friends, heterocyst is a very thick walled structure. Now you understand. So what happens if it is a thick walled structure? Oxygen cannot diffuse. That is what our goal. We don't want oxygen. So because of a thick walled structure, oxygen never ever diffuse. That is one advantage. What is the second advantage, my friends? The heterocyst is lack of PS2. Now you can ask me, sir, what happens when there is a lack of photosystem 2? We already read, when there is a lack of photosystem 2, photolysis of water, that is breakdown of water, never takes place. Friends, if the water breakdown never takes place, which is not releasing, my friends, oxygen is not at all releasing. Simple concept. That is our ultimate goal. So even though oxygen is released, do you think oxygen can cross it? No, because Heterocyst is a very thick walled structure, never ever allow the diffusion of this particular oxygen. Don't forget the statement at all. So this is what one of the mechanism we called heterocyst. Now let's see another mechanism. See friends, I am a bacteria. Like me, many bacteria are involved in the nitrogen fixation. Whatever the bacteria it may be, doesn't matter. They have to maintain anaerobic condition while fixing the nitrogen, otherwise they can't fix. Now let me tell you an interesting point to all of you. Aerobic nitrogen fixing bacteria. You listen. Understand everyone, I am a aerobic nitrogen fixing bacteria, which means if I want to survive, if I want to grow, if I want to develop, I require oxygen. But if there is a oxygen, the nitrogenase enzyme present in my body, it will get inactivated. It can't fix the nitrogen. Then my friends, what is the solution for me? I am a aerobic, I need oxygen, but I want to fix the nitrogen. See how I'm gonna do that. Best example we can call the Azotobacter and Gliotheciae. Azotobacter is the bacteria which maintain reduced oxygen condition through high levels of respiration. Don't confuse friends. In the cellular respiration, what happens? More amount of the oxygen will be consumed. So what the Azotobacter is doing, it increases the levels of respiration. By that, more oxygen will be consumed. So during the nitrogen fixation, in the Azotobacter body, oxygen levels are drastically fall down. When the oxygen levels are drastically fall down, nitrogenase enzyme present in the Azotobacter will become active. Very simple statement. Next one, Gliotheciae. Don't confuse friends. What the Gliotheciae will do? It evolve oxygen photosynthetically during the day and fix the nitrogen during the night. Because during the night, inside the Gliotheciae bacteria, oxygen is not present. Whatever the oxygen is present, it will use all the time during the daytime. In the night time, in the Gliotheciae bacteria body, oxygen levels are extremely low. As a result, what happens? Gliotheciae can fix the nitrogen only during the night time. Do not confuse with that. Next one we called facultative organism. What are facultative organism, friends? Very simple, right? I already told you. They can fix the nitrogen in the presence of oxygen also, in the absence of oxygen also. Then there is no problem. So they generally fix the nitrogen only under anaerobic condition. No problem for us. Anaerobic nitrogen fixing bacteria, it is not at all a problem. What is our goal? Our nitrogenase enzyme should not require oxygen. Even bacteria also should not require oxygen. Then it's not at all a problem. So my friends, what you have to remember now? If any bacteria, whether it is a free living bacteria, whether it is a symbiotic bacteria, doesn't matter. If a bacteria is involved in the nitrogen fixation, while doing the nitrogen fixation in their body, it has to maintain the anaerobic condition. The reason behind that, to activate nitrogenase enzyme during the nitrogen fixation. Simple concept. Now listen. I was continuously saying to all of you that plant and bacteria are going to maintain the symbiotic relationship. Now see here, there is an important line written here. The symbiosis between the legumes and rhizobia is not obligatory. Sometimes these statements will always make us confused. What do you mean by that? Not obligatory, which means, do you think bacteria and leguminaceae plants will maintain the symbiotic relationship all the time? That is absolutely wrong statement. They are not going to maintain the symbiotic relationship all the time, never ever. So when they are maintaining the symbiotic relationship, the question is very simple. They will maintain the symbiotic relationship whenever there is a requirement of the nitrogen. If there is no requirement of the nitrogen, why the bacteria and plant has to maintain the association? They never maintain. Simple point. So that's why symbionts means plants and bacteria. They seek out one another and elaborate exchange of signal When, my friends, only under nitrogen limited condition. Friends, pay attention. Try to remember every single point. Now you can ask me, sir, what is the ultimate goal of the symbiotic relationship? Why the symbiotic relationship has to occur? The ultimate goal of the symbiotic associationship is, my friends, root nodule formation. Very simple, right? That is the ultimate goal. Now from here, every point you try to understand. In this particular root nodule formation, friends, there are many genes are involved. Many genes. Among many genes, there are two important genes are there. One we called nodulin genes, another one we called nod genes. My friends, nod genes, we will be also calling them as the nod factors. Friends, what is the difference between the nodulin genes or nod factors? Friends, whether it is a nodulin gene, whether it is a nod gene, doesn't matter. Both genes are involved in the root nodule formation only. But the difference is, my friends, it present in the different organisms, which means nodulin genes present in the plant only. Of course, they are required for root nodule formation. Nod genes are nod factor. They're present in the bacteria, especially. They also required for the process of nitrogen fixation. There is a no doubt about it. My friends, now 2018 June, this is the direct question they have asked in the exam. Okay, listen, in the part C section for the 4 marker. Nodulin genes are present in the plant. Nod genes are nod factors present in the bacteria. How does this particular nod factors looks like? My friends, please kindly don't worry. You no need to remember the structures at all. Do not worry. Nod factors, this is the first statement they have given in the exam also. Nod factors are lipochitin oligosaccharide signaling molecule. Of course, true. They are made up of, friends, NAG and NAM. N-acetyl glucosamine and N-acetyl muramic acid, which is associated with a glycosidic bond. And they have what kind of a linkage, my friends? Beta 1,4. You know, in our CSIR exam, as one of the option, they have just given alpha 1,4, which is a wrong statement. That is the question they have asked for the part C. Such a simple question. So all of you remember, nod factors or the nod genes present in the bacteria, they are lipochitin oligosaccharide signal molecule. They have a chitin beta 1,4 linkage. Do not forget that. Now friends, what are the type of nod factors? What are the type of nod factors or nod genes? Is there any necessity to remember? Of course. If you look into the question paper of 2015 December, part C question, this is the direct question which we got. There are nod genes or nod factors which are present in the bacteria. There are of two types. One we call common nod gene, another one we call, my friends, host specific nod gene. If you ask me, sir, what is the difference between the common nod gene and host specific? Name itself indicating, na friends, common nod genes are present in every single bacteria. Every bacteria contain the common nod gene. No doubt about it. Host specific nod gene, once again the name itself very clearly indicating host specific nod genes are very specifically present in a particular species only. What are the best example? You have to remember the example. There is no exception here. Common nod genes are nod A, nod B, nod C. Host specific nod genes are nod P, nod Q, nod H, nod F, nod E, nod L. Very simple. P, Q, H, F, E, L, A, B, C. I don't know what you do, friends. You have to remember. Here comes a very interesting point. What is that very interesting point? If this particular common nod gene and host specific nod gene wants to be activated, there will be a requirement of a gene called as a nod D gene. My friends, everyone has to remember, without a nod D gene, you can't imagine the activation of the common nod gene and host specified nod gene. First, this has to be expressed. And you know what do you call it as? We called nod D as a constitutively expressed gene. My friends, what do you mean by the word constitutively expressed? Constitutively expressed means continuously expressed. Nod D is present in my body. I am a bacteria in my body. Nod D is present. Whatever the condition it may be, that Nod D gene has to be expressed continuously. Whenever Nod D is expressing continuously, then only it can express the host specific genes and common Nod genes. My friends, now let me explain you exactly how the root nodule formation occurs. Later, I will solve all your doubts. I am requesting everyone, friends, put your effort, put your attention, try to focus what exactly I am saying. I will explain line by line elaboratively, just pay attention. So, we are talking about how the root nodule is going to be formed. In order to form the root nodule, first bacteria and plant has to be associated with each other. When the plant and bacteria associated with each other, then only root nodule can be formed. Now the question comes here, how does the bacteria and plant come close together first of all? Everyone listen, my friends, listen my statement, I'll explain twice. This is the root hair, correct na? Friends, the root hair of the plant, what the root hair of the plant is doing, it starts releasing certain chemicals. What do you call that particular chemicals? We called it as the chemoattractants. So what are the best example of chemoattractants? 1000% you have to remember the examples which were more oftenly asked in the exams. One we called flavonoids, another one we called, my friends, betaines. Flavonoids and betaines are the name of the chemoattractants released by the root hair. My friends, once these chemoattractants released by the root hair into the soil, they will travel to the soil, they activate the bacteria. The question is very simple. Which gene of the bacteria is getting activated first? Undoubtedly, Nod D gene will activate first. Then after the Nod D gene, host specific Nod gene and common Nod genes are also getting activated. Now you can ask me a question. Sir, this is the root hair, this is the bacteria. How exactly bacteria can come and attach us to the root hair? Now all of you pay attention, listen to me carefully. Now bacteria got attracted towards the root hair. There is no doubt about it. Everyone look into the diagram. If this is the root hair, my friends, on the root hair there are several receptors are there. What do you call it, this particular receptors, friends? Listen, I will explain twice. We called, my friends, lectin receptors. To this particular lectin receptor, who will come and attach us? Bacteria will come and attach us. So friends, every one of you listen to me carefully. The attachment of bacteria, listen, the attachment of bacteria to the receptors of the root hair, we called this particular process as what, friends? We called it as a Rica adhesion. Rica adhesion. For your understanding, I am repeating. Listen to me. First, root hair release the chemoattractants. Example, flavonoids and betaines. Flavonoids and betaines enter into the soil, attract the bacteria. Now all the bacteria come close to the root hair. They will come and attach us to the lectin receptors present on the root hair. Attachment of the bacteria to the root hair, we called, my friends, Rica adhesion. Now you can ask me very clearly, sir, bacteria associated with the receptor, that's fine, but how does the bacteria enter inside? My friends, all of you listen to me very carefully. You can see the picture. Bacteria causing curling of the root hair. What the bacteria causing? Curling of the root hair. Now you can ask me, sir, why it is caused curling? It is causing the curling to enter inside. You can see, friends, beautifully it is entering. So when the bacteria traveling through the root hair, we call it as the infection thread. What do you call, my friends? Infection thread. Now all of you listen to me very carefully, friends. When the bacteria traveling inside the root hair, what happens? Bacteria lose their cell wall. Listen to me carefully. When the bacteria is traveling in the root hair, it lose its cell wall and bacteria multiply rapidly, and it is surrounded by a plant derived structures which we called bacterioids. That will fix the root, that will produce the root nodules. Now everyone look into this. Whomever did not understand, once again read it. plant root releases isoflavonoids and betaines. What did I tell you? They will enter into the soil. They are going to activate the NOD-D gene. Then what the NOD-D gene will do? NOD-D gene will go and activate the another genes. Common NOD gene and host specific NOD gene. Now I have told you very clearly. Now the bacteria will come and attaches to which receptors present on the root hair? Lectin receptor. This process only, my friends, we called RICA adhesion. I hope you are getting very clearly. Now immediately what happen? Once the bacteria associated, they will cause curling of the root hair. Why they cause curling of the root hair? To enter inside. So when the bacteria entering into the root hair and traveling continuously, we can call the infection thread. What is happening now? In the infection thread, bacteria multiply rapidly, my friends. Bacteria multiply rapidly, lose their cell wall, and they can form a structure called, my friends, bacterioids. That is the one which is going to fix the atmospheric nitrogen. Now every one of you pay attention. Sir, why we have done all this? The ultimate goal is what? We produced the nodules. This is our ultimate goal. So what the nodules are going to do, everyone pay attention, look into this beautiful diagram. The nodule contain, what the nodule contain, friends? It contain oxygen binding protein. Oxygen binding heme protein. What do you call, my friends? We will be calling it as the leg hemoglobin. Now friends, what does the leg hemoglobin is going to do? We commonly know that the hemoglobin give rise to the red color. But friends, since the leg hemoglobin is present in the infected plant cells, infected nodules, it will give pink color. Now you can ask me one straightforward question. Sir, what is the ultimate function of the leg hemoglobin? My friends, the ultimate function of the leg hemoglobin is it is having the highest affinity for oxygen. That is our goal. Friends, if leg hemoglobin will go and associated with the oxygen, then oxygen level becoming what? Low. When the oxygen level become low, who will get activated? My friends, undoubtedly nitrogenase enzyme is getting activated. That is our ultimate goal. If I want to fix the nitrogen as a bacteria in my body, nitrogenase enzyme should be active. For that reason, once the leg hemoglobin is produced, it will go and block the oxygen. Then nitrogenase enzyme become active. This is the reason, friends, the symbiotic relationship has to occur. Now, ultimately, friends, we have produced the nitrogenase enzyme and nitrogenase enzyme become active. Now, we are going to see the real step. Once this step is over, I'll solve all your doubts. Now, what is the major step? Nitrogenase enzyme is ready. Now what we have to do? We have to fix the nitrogen. Everyone pay attention, try to understand every single point. So there is an important gene. The gene which is involved in coding for the nitrogenase enzyme, we called NIF gene, NIF. Name of the gene is what? NIF gene. What is the NIF gene? Listen my statements, every one of you. Whatever the enzyme it may be, whatever the enzyme that is involved in the nitrogen fixation, all those particular enzymes, all those particular proteins can be coded by this particular NIF gene. Don't forget the statement. Now friends, ultimately we have activated the nitrogenase enzyme. Now let's complete our goal. What is our goal? I hope all of you remember. This is the goal of nitrogen fixation. Conversion of N2 to NH3 or NH4 plus. This is our goal. Let's fulfill our goal now. Before that, I want every student has to understand nitrogenase enzyme structure. Sir, will they ask this in the exam? Undoubtedly. When in a given 4-marker question, at least one statement they will rise with respect to the structure. Then it is idealized to remember the structural properties without fail, friends. So the nitrogenase enzyme contains 2 subunits, dinitrogenase reductase and dinitrogenase. My friends, just remember like this. Dinitrogenase contain iron sulfur redox center and molybdenum iron redox center. Just remember like this. Iron sulfur and molybdenum iron. Friends, from the starting of the lesson, I was continuously and repeatedly saying that nitrogenase enzyme will get inactivated if there is a presence of oxygen. But why? The reason here, The nitrogenase enzyme contains 2 subunits, right? Dinitrogenase reductase is the subunit which is sensitive to the oxygen. That is the reason we are repeatedly saying nitrogenase enzyme will become inactivated by the oxygen. My friends, now the nitrogenase is ready. Now we are going to fulfill our goal. But my friends, every student has to listen what exactly I am talking for the period of 10 minutes. This 10 minutes of understanding is very important. Very important, pay attention. Listen to me. What is my goal? This is enzyme. What enzyme? This is the nitrogenase enzyme. To the nitrogenase enzyme, who will come and attach us now? N2. So here N2 can act like what, my friends? Substrate. Correct na? So I have to convert this N2 to NH3 or NH4 plus. Friends, everyone pay utmost attention. But I can't fix the nitrogen directly into NH3 in one step. That's a difficult. This can be done in 2 steps. What is the first step? In the first step, N2 can be converted to, my friends, NH double bond NH. Do not forget, my friends. Then second step, NH double bond NH can be converted to, friends, NH2 and NH2. Then finally, friends, NH2 NH2 can be converted into NH3. So my friends, I can't do the process in a single step. I require 3 steps. And here comes the most important point. Friends, every step to occur, we require 1 pair of electron. So here we require 1 pair of electron. Here we require 1 pair of electron. Here we require 1 pair of electron. So my friends, listen to me carefully. But one more interesting point. When the N2 converted into NH3, don't confuse. When the N2 can convert into NH3, first N2 has to attach us to the nitrogenase enzyme. Listen to me carefully. If nitrogen acting as a substrate molecule want to attach us to the nitrogenase enzyme active site, it require another pair of electron. So total here 1 pair we are using. 1 pair for what? Binding nitrogen. Then conversion of nitrogen to NH3, we require second pair, third pair, 4 pair. Total 4 pairs we require. 4 pairs means total how many number of electrons? 8 electrons. Sir, why you are saying about electrons? Now listen, that is very important. Because this iron sulfur redox center contains 2 ATP binding sites. Friends, what do you mean by the word 2 ATP binding sites? Which means, listen to me everyone, if one electron passed through this iron sulfur redox center, it will hydrolyze 2 ATP molecules into 2 ADP molecules. If I want to convert the N2 to NH3, total how many electrons I required? 8 electrons I required. So 8 electrons when they pass through the iron sulfur redox center, 8 into 2, how many molecules of ATP we are using? 16 molecules of ATP. That's why in a given 4-marker question, they will give one statement. Nitrogen fixation is a highly energy consuming process. Absolutely true statement. You only try to understand we are using 16 molecules of ATP. I will re-explain to all of you. Pay attention. It's easy. It's easy. This is the structure of nitrogenase enzyme, my friends. Contain iron sulfur protein, molybdenum iron protein. What is my goal? Just listen to me. My goal is what? Conversion of N2 to NH3. This is my goal. For this, how many steps are required? 3 steps are required. Now, I was saying we require electron. Now, the point every student has to remember, who is the donor of electron? The electron can be donated by ferredoxin. Very simple, my friends. First, ferredoxin donate the electrons, 1 pair of electron. For iron sulfur protein to molybdenum iron to electron pair comes to N2. When electron pair comes to N2, it will convert into NH double bond NH. My friends, another pair of electron is coming once again. From iron sulfur protein, molybdenum iron protein, then where it will come? It will come to NH double bond NH. Now it is converted to NH2 NH2. Once again, third pair of electron is coming. Once again from iron sulfur, molybdenum iron, where it will come? To the NH2 NH2. My friends, undoubtedly NH2 converted into NH3. Very simple point. So here we use 3 pairs of electron, and to attach our nitrogen to the nitrogenase enzyme we use another pair of electron. So total 4 pairs, 8 electrons. Now 8 electrons are hydrolyzed and give rise to how many? Sorry, when 8 electrons are passing, 1 electron involved in the 2 molecules of ATP hydrolysis. Then we will hydrolyze 16 ATP. Now see here, the same thing I have written elaboratively. First, nitrogen bind to the enzyme complex. We require 1 pair of electron. Who is donating electron, my friends? Ferredoxin is donating. From ferredoxin to electron comes to iron sulfur protein to molybdenum iron protein. Then N2 converted into what, my friends? NH double bond NH. This is the first step, simple step. Then the cycle repeats twice more. Then in the second step, we produce NH2. Third step, we produce NH3. My friends, now let me summarize the equation. You can only tell me nitrogen, 8 electrons anyway we use to maintain the charge balance, 8 protons. Nitrogen, 8 protons, 8 electrons. We spent 16 molecules of ATP. Then my friends, N2 is ultimately converting into NH3 by the release of hydrogen, and 16 ATP hydrolyzed to 16 ADP. Friends, this is the first step of nitrogen fixation. I hope it's clear. Now you can ask all your doubts. Is it clear, friends, for every one of you? I hope we did not go fast, but I explained briefly. Is it clear for everyone? Yes or no, friends? You can ask any kind of a doubts. All of you. All people. Okay. It's a very simple process, friends, and easy process. Okay, that's great. Okay, I hope everyone understand in a clear, sir. In each cycle only single electron passes or pair. Mrunalini, always dear, pair, one pair, which means 2 electrons. One pair will go, dear. Okay, that's great. I hope everyone understood very clearly, friends. Remaining part we will do in the next class. Now everyone, please give me SECA is similar to CDC6. No. J3, that is not the answer. SECA is inhibitor, dear. CDC is the cyclin. That's a wrong statement. Frankia fixes nitrogen in the roots of alnus plants. You mentioned something different. No, dear. No, Rins. I did not mention anything different. I have given right. Alnus also it will be, and I did not mention anything differently. Okay. Alnus I did not mention. That's all. It is alder, rubber tree. In rubber tree also Frankia will fixes. It's not different. In rubber tree also alders are rubber. Okay, you got it. Okay. So friends, now everyone please answer my question. Can I start from tomorrow morning? See, if I want to start and if I want to do something for you people, first I should be very disciplined. So if you give me the commitment that you will be there with me for a long time, then only I will take my responsibility and I will start the classes from early morning 5:30 to 7 a.m. instead of evening time. By that benefits are you can wake up early, you can revise, your brain is also very fresh and you can be motivated all the day, you can prepare. But if you assure me that you will be with me for last till the June, see, if very less number of students are attending, see, if one fellow is there, if 50 fellows is there, I will teach in the same way. But when more people are there, the engagement is different. Everyone will get interest. Okay, if everyone assure me that till June you can attend the classes 5:30 to 7 a.m., I will start from tomorrow morning itself. Okay, Jayashri, I understand, dear. Degradation of CDC6 prevents reinitiation. Okay, dear, that Jayashri, that also a true statement, dear. But CDC cannot act like a SECA. Maybe it is having a different mechanism, dear, but CDC cannot act like a SECA. Friends, everyone, if everyone give the answer, if not 2 to 3 people, if everyone is ready, that's what Tarun, instead of evening, I will spend 5:30 to 7 in the morning itself. That's a 1.5 hour is a big time. We can cover many things. Oh, that's amazing. Okay. I hope everyone is ready. Even I am also ready. Okay. So let's start from tomorrow itself. Sharp 5:30 class will start. Okay. Till 7 a.m. Okay. It's my responsibility in the Telegram group. I will give the message at sharp 5 a.m. You people just get ready. Okay. We will go very faster and we will do very effectively. If I have 1.5 hour of dedicated time, I'll cover more concepts in a fast, in a little effective way. Okay. Okay friends, that's all for now. I think all the videos and all the PPTs are already uploaded in the website. You can download from there. Okay. Okay friends, that's all for now. I'll see you tomorrow morning 5:30 a.m. Bye bye. Thank you so much. Good night all of you. Good night.